Welcome to the RahbarTalkShow. Here we want to continue our last discussion topics. It was related to protein corona. And so because of its complexity and intricate mechanism which are involved in decoration of the exosomes, nano-sized bioshuttles, so we want to continue our discussion in this session. Please join us and see what happened. Mrs. Shabkhizan, as you know, some studies reveal that the formation of the protein corona can just lead to some detrimental effect inside the cells after internalization of protein coronated exosome. Do you have any data about it?
One of the most important challenges that we face in utilizing nanoparticles in medical applications is cytotoxicity. There are some key factors.
Coronated exosome. You mean it has some cytotoxicity? Am I right?
Yeah.
Great.
Exosome that coated by protein corona, actually. And there are some key factors that affect this cytotoxicity that we will discuss it in this session. But in all of the situation is not constant, and this effect can change in some different situation.
Great, thank you. All in all, you are believing in fact that the protein corona has its own detrimental effect on the target cells after internalization of the decorated exosome.
Yeah.
And so, how do you think about the beneficial effects? Maybe that just exosome just absorbs some of the proteins which are beneficial to the metabolism of the target cells. Can it be true?
Yeah, is it right? But
you haven't enough data about it. Yeah?
No.
Okay,
yeah.
Let's see what happened. Yeah, Mrs. Salimi, can you help us to just scrutinize which kinds of the mechanism are involved in the detrimental effects of the decorated exosome, that the protein-coated exosome inside the cells?
Yes, the formation of protein corona can enhance and suppresses nanoparticle-induced cytotoxicity. This effect is highly dependent on protein composition and nanoparticle surface properties. For instance, proteins such as albumin, fibrinogen, and immunoglobulin can promote uptake by certain cells, thereby increasing intracellular interaction and RAS production, reactive oxygen species. Activation of MAP kinase and ER kinase, and in addition to PI3 kinase pathway, has been reposted in response to nanoparticle and protein complex.
You mean that the protein corona compositions and the composite can just directly they interact with this effector, intracellular effector for MAP kinase and other signaling pathway molecules.
Yes, this led to
the alteration of the metabolism, even that acceleration or inhibition.
Cell survive, proliferation, and apoptosis. Additionally, complex of protein corona and nanoparticle can induce mitochondrial dysfunction and lysosomal permeabilization.
In that case, some of the enzyme and hydrolases can just leak to the cytosol and just promote some digestive reactions.
Yes, that's right. We are the protein-mediated endocytosis.
Oh, great, thank you. Thanks for your explanation.
So, Professor, I would like to add some comments along with my friends, as mentioned,
Please.
about the protein albumin and the protein corona composition, namely sometimes albumin, serum albumin, can decrease the surface energy by this way, and also at the same time increase the cell membrane interactions. So this means that the interactions with the cells is going to increase, and by this way the cell, the nanoparticle uptake is going to be enhanced. And these things that I would like to mention, and also another things, the cell types is so important here. Some cells are going to these protein coronas going to have a positive effect on them and profound effect. It is going to increase the cell uptake. On the other hand, some cells is not going to act like this and decrease. So this is the debate, ongoing debate, and also some more research needs in this area.
Yeah, absolutely. You're right. You are exactly right. Based on the types of the proteins which are absorbed on the surface of the exosome, for example, transferrin or other macroglobulins. There are kinds of the serum proteins which can be just randomly, I don't know, can be just absorbed to the surface of external surface of the exosome, and they can have their own receptor on the cell membrane if they are membrane bonded, and so that can, yeah, absolutely can activate the cell entrance. But otherwise, it's also possible that there's some kinds of protein and covalent interaction, as we discussed the last sessions, can just cover some the heterosponins or other ligands on the exosomal ligands, I mean. And so it can just maybe just postpone its entrance into the cytosol.
Yeah, and this happened in the, I think that according to some research, it happens on the surface of the outer layer of the protein corona, namely called soft corona. And this is a very environment dependent area of the protein composition. And by this way, it is going to exchange some materials with the serum medium. And by this way, yeah, this happens.
It is also possible that some of the covalent interaction occurs on the protein corona layer, which could not be digested and analyzed by the lysosomal degradation. So it can just last for a long time. And this is some kinds of the toxic materials. But the mechanism and the pathway which are involved in toxification of the host cells are not fully addressed, and so have not been fully addressed. And so in the future studies should just focus about addressing this issue. Okay.
Sorry, Professor. I think
You're welcome.
there's, I've read some articles and research that has some contrast results with whatever talked here. Actually, there are some environmental factors which are really important in the, how to say, stabilizing the protein corona or even getting bigger or getting smaller. These two factors are the temperature and the pH, which means that if they have really, really diverse effects on protein corona complex, which may stops it and decrease the amount of protein corona, which leads to more absorption, and that leads to toxicity. But also there are some other results that says that actually when these factors, I mean the pH and also the temperature of the environment decreases, the protein corona complex gets bigger. It means that it easily absorbs as a surface of the nanoparticle or exosome or anything that which is trying to get into the cell. And when the outer layer and when the inner layer thickens, it means that the toxicity goes down and it stops the absorption and really affects the process. So the pH and environment temperature is really important because it has some physical effects. It's not always, it's not chemical.
And you mean that just by increasing the size of the hard core, maybe hard layer and soft layer, its toxicity is going to be altered, yeah?
To make a long story short, yeah. When the protein corona thickens, the toxicity goes down because it stops from the absorption.
Do you think that it's, I don't know, the toxicity is the closer related associated with the type of protein, not about the amounts of the protein?
Actually, yeah, I've mentioned that it's the physical view about the protein corona, not the chemical or not the ligands view. I mean, we are just watching it as a physical complex. When it gets bigger, the absorption goes down, so the toxicity goes down. Actually, there are lots of results, different kind of results in different research, and I just want to mention that it's not that easy. We think that, yeah, it gets bigger, it doesn't absorb.
Yeah, absolutely.
Yeah, it
That's
goes
a big,
smaller,
yeah.
it absorbs. They have lots of factors. I just mentioned about the physical view of the complex of corona, protein corona, around the nanoparticle. Of course, there are chemical effects. Of course, there are molecules that are related. Of course, I'm in the same ground with you about the kind of the proteins which are covering the nanoparticle, which is really important because of the ligands, because of the markers, because of the connectors. Yeah, of course, but physical view, temperature and pH is really important. And the size. The size does really matters. It gets bigger, the absorption goes down. And it is in the same ground as in the former session I mentioned about the Lipinski's rule of 5. Because the weight goes up, the absorption goes down. This is something which is a fact. When it gets heavier in weight, the absorption goes down.
Great.
I have a question in this
Yeah?
regard from you, and this is right now.
It's related about this.
Yeah, it happens in my mind, and I would like to ask this question. If the size is going to increase, and maybe the targeting is going to be altered, and it is not going to directly received by the targeted cells, and in other part of the body, for example, spleen, it is going to be accumulate, and the risk of the tumor may be increased. This is a question in my mind, and I would
Actually,
like to
just directly related
ask
to just anaplastic changes. I mean, that tumor formation effects. I think that the change in the size can just lead to the reduced retention time. You know, because I know absorption, different proteins with different function can just assimilate the different cells to just uptake these particles, and it's going to just lead to the shorten the lifespan in the circulation system rather than the tumor formation effect. Because as you know, exosomes, in contrast to the cell source, I mean that the cell transplant, cell source which are used for transplantation and regenerative purposes, could not just induce the anaplastic changes, as I know, except the oncosome, the tumor cell-derived exosome. We called it the oncosome. So it is mighty, and it is not mighty, and so the possibility is not high. So we just considered, we just mainly focus about the detrimental and cytotoxic rather than the anaplastic changes. You know what I mean? Yeah. Okay, Mr. Shakwiza, any comments related to? You're welcome.
Actually, you are completely right about the effect of temperature and pH on nanoparticles, but consequently, it affects, as you said, in the size of the nanoparticles. And as I said, the size of nanoparticle is so important in cytotoxicity because based on scientific evidence, the smaller nanoparticles can easily internalize to the cells and even some organelles like mitochondria and even nucleuses. So it can affect some cancerous cells and some
normal cells.
Some normal cells, yeah. So size is so important in cytotoxicity.
Yeah, actually, I mentioned that it's a physical point of view about the protein corona, not the chemical view, because even temperature, even pH, or any other factors which are related to protein corona, yeah, actually, as a result, we have something with a size and weight. So, yeah, that's right.
I think it's also possible that increasing this mean diameter of the decorated exosome can change its internalization mechanism, you know. For example, imagine that some of the proteins are adsorbed to the surface of exosome. So, as you told us, its diameter will be increased. So, it is also possible that these exosomes can just enter to the target cell via, for example, phagocytosis or other mechanism, rather than ligand and receptor-based mechanism. So it is possible, yeah.
May I add something?
Yeah,
also, the shape of the nanoparticle here is important too. And I think that according to some results and studies, protein corona is going to reshape the nanoparticles. Some nanoparticles, such as the nano gold rods, or the some have a bad angle. I mean,
We called it allosteric changes. Allosteric
Yeah.
changes in molecular shaping, and so it's going to breed.
it is so
some-
sharp,
Yeah.
and it is going to hit other cells. By this way, by reshaping that, it is going to be smoother, and smoother nanoparticles is not going to be toxic any for the other cells. And from this view, it can be also helpful.
Yeah, you are exactly right. We all just focus about the target cells after internalization. I mean, that there's some the cytotoxicity inside the cells, but it's also possible.
Professor, I can a question. Can you explain a little about the signaling of reshape of nanoparticle protein corona?
Yeah, as we told previously, these some kinds of proteins, for example, complementaries or fibrinogens or other molecules which are adsorbed to the surface of exosome, they can just, provoke the activation of the some intracellular molecules and effectors. So as you told us, a MAP kinase or other kinases can just be activated, and so the cascading molecular cascading will be initiated. Yeah, absolutely. As I told, we just mainly focus about the cytotoxicity inside the cells, but it is also possible that the absorption of some proteins to the surface of exosome can activate coagulative factors. I mean, that inside the blood. For example, complement or fibrinogens, which are adsorbed to the surface of the exosome, they can just activate these molecules. So the coagulation cascade can be just stimulated, and so some pathological condition can be also happen. But we should just focus how these molecules can just activate, and with types of the biochemical reaction which are involved should be addressed by future studies.
Excuse me, Professor.
You're welcome.
One of the other key factors that is important in cytotoxicity is surface charge. And to set an example, one study that was established in Italy recently, they examined the effect of protein corona and cell surface charges on cytotoxicity in breast cancer cells. And this study indicated that, for example, the material of nanoparticles is important. For example, in silver nanoparticles, cell surface charge is more important than protein corona and have bigger role. But for example, in some kind of cells like immune cells and stem cells, the story is different. And in this kind of cells, protein corona's role is more important and bigger than cell surface charge.
Great. Yeah, you are right. You are exactly right. Depending on the functionality of each cells and different mechanisms which are involved in their metabolic activity, each cells can be sensitive to different aspects of the protein corona. Even that there's some charge, even that the compositions, and even that the diameter, some other parameters are so important. That should be just considered. Thanks a lot.
Professor, I have a question about these recommends. How we can suppress some negative interaction between protein corona and nanoparticle, for example, in cancer, breast cancer?
Actually, this is out of our control. The inhibitions or regulation of the nanobio interaction is somehow challenging because of its complexity and intrinsic mechanism and different reactions. Even the physical and chemical interaction occurs between the decorated exosome with the host cells. It is actually impossible at the present time. So the sophisticated technique should be just considered. Even that I would just read an article related to the separation of the protein corona from the external surface of exosome using some high solute solutions. This should be, relatively, in part, a strategy to just remove the protein corona, but I'm not really sure about it can work or not. And so we just be patient and see what happens in the future. Fazin, as you know, that the formation of protein corona around each nano-sized particles can cover its surface. And so different molecules which are naturally or adhere to the surface of the exome also be covered. And do you think that it can just influence any interaction of these exome with the target cells? I mean, that it can alter the delivery efficiency or not?
Yeah, absolutely. Everything affects the nanoparticle, even the protein corona complex. Actually, I'm looking from the negative side. And as I mentioned in the former speakings, actually, yeah, the increase of the size is a real big problem. So there are some methods that they are working on it. Actually, it's not proved completely. There are some results. They have some contrasting results, good results, bad results. But there are two factors which really enhance the size and absorption and also the targeting of the protein corona. Actually, the protein corona complex itself has negative effect in targeting the nanoparticle to have intracellular interaction or not. So if we are looking from the negative side, which it stops the nanoparticle to easily absorbs to the cell. So what they do, they add some molecules like polyethylene glycol, and also there are some zwitterionic ions like cysteine, which when they add these molecules to the protein corona complex, it reduces the size and effects of protein corona against absorption. So from the negative side, yeah, it's controllable sometimes, but still needs lots of work. We are not sure about it. It's not written, or it's not like physical factors, which is important, but they are working on it. And also they alter some surface proteins, which is really important. And also it goes back to physical appearance.
I'm so sorry to interrupt you, but any manipulation per se can just also alter the further protein corona formation. I mean that it's going to just change the composition iatrogenically, because we're going to alter it and we're going to manipulate it.
Yeah, sure. Actually, they are manipulating it by adding polyethylene glycol and cysteine.
As I know, it has some determined effects on the integrity of the exome, because polyethylene glycol is going to just permeabilize exome, and just so the cargo will be the cargo of that exome can just leak to the outside, unfortunately.
It has some amount that they are using it. It's not like that destroying the whole complex. And they just manipulate cysteine and PEG in a way that it just decreases the size of protein corona complex, not in a way that it destroys the whole package.
How about the net charge? Yeah, please.
I would like to add some comments according to the my friend mentioned that polyethylene glycol, the use of the polyethylene glycol, shortly PEG. This is belongs to this dependent to the physicochemical chemistry of the properties of the nanoparticles, because by this way the hydrophobic interactions is going to be low. The less the hydrophobic the nanoparticle is, the less it is going to have interactions and some bonds between the proteins, and the less proteins is going to absorb on the surface of the protein nanoparticle. By this way, I think that this is maybe right now, this is one of the solutions in order to reduce the absorption of the protein corona, in order to have some positive effect of the nanoparticles when we're talking about the
but to be honest,
targeting therapy.
To be honest, because of the enormous proteins and different biomolecules are adsorbed at the surface of the exome, I think that any manipulation per se can contribute to any post-complication, further complication, post-complication. Yet I'm not really sure about it. But it deserves some strategy.
Yeah, of
just
course.
impart control.
But also, let's reimagine the situation and just think, as we are just want to, for example, deliver some anti-cancer drug or a DNA or RNA into the exact type of cells. And in that case, I think manipulation really, it's needed, important. Because, the only result we want is to have more absorption, to target directly. Of course, I agree with any manipulation will have some side effects. But imagine a situation that we want to just deliver a special kind of drug into tumoric cells. And the only way is, for example, to use cysteine to make the complex smaller, just to have more absorption, to have a targeted delivery. In that case, I think manipulation is needed. But yeah, side effects, I totally agree with you. We will have, because it's a phenomenon. Yeah. Of course, changing something is like a chain that will lead to more problems or maybe some side effects that we are not still aware of it.
Yeah, absolutely.
But,
domino reaction.
Domino reaction. But in targeting, I think it's a little bit logical.
Thank you.
To manipulate.
Thank you for your comments. Very fruitful comments. Thanks a lot. Mrs. Shaprizan, any positive effects about the protein corona?
Actually, recently I read an article about this kind of manipulation in order to targeting nanovesicles. This study was published in Nature Communication recently. They developed some nanovesicles, some special nanovesicles that coated by sugar-based coating. I mean, glycolysated nanovesicles that they wanted to deliver some drug to cancer cells. And it indicate that this kind of coating prevents some immune proteins like CD44 that is important for survival of these nanovesicles in the blood. And they indicated that in this case, these nanovesicles can deliver to the cancer cells very easily.
By modulation of CD44, you mean, yeah? Am I right? 44?
No, this kind of sugar-based coating can prevent this immune protein onto the nanovesicles like CD44 and
it's a kind of cellular protein. Yeah, it's a kind of a cellular protein. These proteins participate in attachment and some physical interaction, as I know.
Yeah.
Okay. Yeah. Please.
Yeah, as I said, it can increase the
internalization of that decorated exosome. Yeah.
Yeah, internalization. Okay.
Thank you. Hesam, as you know, some previous studies have revealed that the formation of the protein corona can reduce the retention time in blood. This is because of the enhanced penetration or internalization of the decorated exosome, I mean that the protein-coated particles, into the immune cell, which are resident in embryonic tissue and liver. Do you have any idea about it?
Yeah, actually, this is structure question, and you mentioned it. There's a lot of things that we can say, but make it short and sweet, actually.
Yeah,
and the important point. I can add more information in regard with your sayings. Absolutely, it depends on the soft corona or soft corona protein or
hard
part. But this can affect because the shape and structure of the protein that decorate for example, exosome is different type. For example, if we can combine with nanoparticles also, it will affect in another way. But in our blood circulation, it can cause to making activate inflammatory signaling pathways or starting the cascade of cytokine storms in our immune cells, actually.
I'm going to interrupt you, but we should be so cautious about how these particles can just inhibit or stimulate some, I don't know, the signaling cascade inside the blood.
Yes. Very briefly and very in the important way, the toll-like receptors, they can actually the corona protein, they can produce toll-like receptors and
activated.
Yes, activated and promoted their function also, and it can affect very significantly, and also. TNF alpha, the activation is very largely in large scale they can affect, and in the phagocytosis they can enter inside the cells, and the story will happen. For example, the how can I say our immune cells response will very suddenly look like starting a very rapid cascade of this, and I'm
Sorry,
going to.
I'm going to interrupt you as I got to understand. There are by your viewpoint, there are two main mechanisms which are involved in the activation of the immune cells. The first one is related to the activation of some receptor, membrane-bonded receptor on immune cell, for example, to like receptor signaling pathway, and even that TNF alpha signaling pathway, something like that. And the other side is that the story starts after internalization of the decorated exosome. Some reaction inside the immune cell, and some of the cytokines and pro-inflammatory molecules are going to be released to the
Yes, for
outside the
such
immune
a
cell.
macrophage. You mean
Macrophages,
that?
yeah.
Yes, macrophage activities, and another white blood cell activities, they will increase, and by increasing them, the inflammatory actually response will increase. By these effects, I can tell a little bit more about this, because this is the unknown actually effect. Because we can't control actually it.
I think that we should just, I don't know, consider the opposite effects as well, because it is so impossible that some of the inhibitory molecules can just absorb to the surface of exosome, which exist in protein coronal composite, and so they can just maybe, I don't know, act as inhibitory molecules.
The interesting thing is in this regard, because of that, in the laboratories, the scientists or researchers try to combine this with nanoparticles to reduce this effect.
Okay, thanks a lot.
Yeah.
Thanks a lot.
Can I ask some?
Yeah, absolutely. Why
about
not?
the
Go ahead,
immunity?
please.
I think the protein corona plays a pivotal role in immune recognition. Corona-enriched nanoparticle often display some protein, opsonin, for example, complement protein C3 and C4b and IgG, which mark this complex protein and nanoparticle for clearance by macrophages.
This example will work on the some of the nanocytes, infectious particles, yeah? But it's not good for exosome, as I know, because it's going to increase their internalization and per se reduce their retention time in blood. Yeah, you're exactly right. You mean that this particle is going to be opsonized, and so the opsonization will increase their internalization into the immune cell. Am I right?
Yes.
Yes. Okay.
Increase the permeability of the cellular surface.
Yeah, absolutely. These molecules
by discharging them.
Yeah, absolutely. They act like a chemotherapy.
And it is bad, and and the scientists try to solve this problem. Actually,
Okay.
we don't need the cytokine storm cascade.
Yeah, absolutely. Thanks a lot. Thanks a lot.
Thank you.
Student, thanks for your presence in this discussion session. Actually, protein corona with complexity and inherent intricate mechanism should be just monitored and studied and assessed by different sophisticated technique. We are not sure how this phenomenon can just affect the bioactivity of different nanosized particles. So I think that it does necessary to just debate more and more about these biological effects, and it should be just, discriminate in different issue. The detrimental effects and beneficial effects. It is also possible the protein corona formation can provide us some of the beneficial outcomes related to the nanosized infectious particles. But how about the exosome? It should be just assessed by numerous experiments, even preclinical and clinical studies. Students, thanks for your presence. It was really very interesting topic, and you just helped me to just reveal some aspects of the protein corona. Thanks for your presence again, and I hope that it will be beneficial to our audience as well. Dear audiences, thanks for watching our video. Please like, subscribe, and share the video, and also write down your comments in our channel. Thanks a lot.